Bismillahirrahmanirrahim. This is the topical MCQ on the chapter of transport and plants, which is the seventh chapter in 9700 AS Biology. Now, starting with question number one, which row is correct for the phloem sieve tube element? Now, the structure of the phloem sieve tube element is very different from the phloem. Phloem is from the xylem. Xylem only has the cell wall, and then it has lignin deposited in it. But the phloem has a cell membrane. It does have cytoplasm, but there's no nucleus, and as there are no nucleus, so there's no DNA, so no protein synthesis can take place, so no ATS ribosomes. You see, the DNA transcription must take place in the nucleus from DNA to mRNA, and then the mRNA comes and gets attached to the ribosome. So if there's no nucleus, there's no DNA, why should there be any ribosomes? That makes sense. Question number two. An aphid such as greenfly uses a tubular mouthpart called a stylet to feed on nutrients in the plant. It inserts the stylet through the surface of a stem or leaf. When a stylet is cut near the aphid's head, leaving the stylet in the plant, a liquid with a low water potential continues to flow out of the plant. Which statement explains this? Now, there's a lot of clues which you're getting in the question. It says inserts the stylet through the surface of a stem. Now let's look at a stem. Stem's got the xylem and the phloem on the periphery, on the outside. And the first thing is that the phloem is on the outside. So when the aphid puts its stylet in, it's going to go in here. Stylet is a tiny tube, and this is the aphid. So it's going to go into the phloem. So these were both wrong. If you know the structure of the stem only, then you can do this question. So the stylet is in the phloem sieve tube, in which there is a high concentration of solutes. You know that the phloem contains a lot of sucrose and amino acids and in water, ions. So this is what you had to know about this part of the question. Now, question number three. Which statement correctly describes transport pathways in a dicot plant? Now, they're talking about the two pathways, apoplast, and I always say that remember apoplast. Apoplast is along the cell wall or through the cell wall. So if these are cells, the apoplast pathway is along the cell walls, through the cell walls, because the cell walls absorb the water. So it will go through the cell walls. This is apoplast. And symplast is cytoplasm to cytoplasm. Because here you would be having the vacuole inside it. So cytoplasm to cytoplasm, and there's a Y in cytoplasm and there's a Y in the symplast pathway. So that is why the answer C was correct, is the apoplast pathway may be blocked by the Casparian strip. So the Casparian strip is the layer of suberin around the endodermis. So that actually is on the outer surface, so that's why that blocks the apoplast pathway. And coming to question number 4, the diagram shows 4 monosaccharides with the formula C6H12O6. Which diagrams do not show glucose molecules? And that's 3 and 4. And I've told you the mistake in them where that was wrong. Question number 5. Which processes are involved in the mass movement of water through xylem vessels? Hydrogen bonding between water molecules, cohesion between water molecules, and adhesion between water molecules and lignin. So it was all 3 of them, 1, 2, and 3. Then number 6, the diagram shows transverse sections of parts of a plant. In the transverse section, which tissues transport most mineral iron? So that, of course, is the xylem. So you can see in the leaf, it is the leaf upper xylem. Leaf upper xylem. So the upper parts are the xylem. So 1 is the xylem. And in the stem, the xylem is on the inside. So that had to be 1 and 4. So this was correct and this was correct. Question 7. Which changes in the water potential and the volume of liquid in the phloem occur when amino acids are transferred from leaves to be transported to a sink in the phloem sieve tube element? You see, if the source is the leaf, so then amino acids, when they are put into it, this will lower the water potential. But what will happen to the volume of liquid? Naturally, if amino acids are added to a sieve tube, then water is going to follow, because the water potential decreases and water will flow in. So the volume of liquid in the phloem sieve tube will naturally increase. So that is what we have to understand, how this is a lower and an increase answer. Then question number 8. External factors change the rate of transpiration, which also affects the water potential gradient of cells in the leaf. Important word, water potential gradient of cells in the leaf. Which combination of statements is correct? Now, if you look at the answer, why is the answer C correct? Now, if there's a leaf inside, there are 50 water vapors, and outside there are 100. Well, water vapors are not going to diffuse out. So if they are not going to diffuse out, this 50 is going to become 90. Now, first the gradient was 50-100. First the gradient was 50-100. Now the gradient is 90-100. So the gradient has decreased. It says water potential gradient in the leaf. 50-100 or 90-100 now. So that's how the answer has to be understood. Then question number 9. The graph shows the diameter of a tree trunk at different times. Diameter of a tree trunk. This is the diameter. So this would be the diameter from here to here of a tree trunk. So we have a tree, and this is the tree trunk, and we're talking of the diameter of the tree trunk. And this is arbitrary units, and the timing is 2400 hours, then 1200 time of the day, and then 2400. So we know 12 is noon, and then of course we have another 12, so 12 at night, and then so at X, Y, and Z, which statement is correct? X shows expansion of the trunk as water fills the xylem during transpiration. Y shows a reduction in diameter of the trunk as water is lost from the xylem due to transpiration. So, which statement is correct? They're just talking to you about the diameter of a tree trunk at different times. So you can see the diameter decreasing at 1200 hours in the time of the day, which is more of the noon time. And then, so at Y, there is a reduction in diameter. So we were only going to consider these two different answers. Why? Because X shows the expansion of the trunk. We're just showing the diameter of the tree trunk is increasing at X and at Z as well. But the answer to that, why that is decreasing or increasing, is wrong. So that is why the answer to this was the B part of the question. The B part of the answer was correct. Y shows a reduction. Diameter as water is lost from the xylem due to transpiration. As time of the day, stomata open, increasing light intensity, more the rate of transpiration. Question 10. Some plant species can tear up toxic heavy metal ions that are dissolved in water in the soil. These ions accumulate in the aerial parts of the plant, which can be harvested and disposed of safely. An investigation was carried out into the ability of a plant species to remove a heavy metal ion from the soil. This produced the following observations. Some heavy metal ions accumulated in the cells of the root cortex. Within these cells, most of the ions were in the vacuole. So cell, but they were found inside the vacuole. In the roots and leaves, the ions were transported dissolved in water. Which suggestions about the transport and accumulation of heavy metals are valid? So you read these once again and then you look up what is valid. After initial entry into the root, some of the ions can pass through the tonoplast because they were found inside the vacuole, so that means they can pass through the tonoplast. In the roots and leaves, ions pass through a symplast or an apoplastic pathway. But as the endodermis, they must take a simplest pathway, but that's only for water. In xylem vessels, the ions pass through an apoplast pathway. Apoplast pathway is along the cell wall or through the cell wall. So that's why the answer was C, because this was wrong. Both these were wrong. The diagram shows the results of an experiment on transpiration using two different leaves. Two different leaves. Each leaf was left on a balance in a daylight in a closed room, and the mass recorded at 1 hour intervals. So there's 0 hours, then we have 1, then we have 2, and this is the time in hours, and then this is the mass of the leaf. So as I always say to you, like in the enzyme question, also put some figures on it. So say this was 5 grams. So leaf 1 was also 5, and was leaf 2 was also nearly something 5 or less than 5. And then we see their mass decreasing. So if say this was 5, then it's 4, then it's 3, then it's 2, then it's 1. So leaf 1 went from 5 to say 3, lost mass from 5 grams went to 3 grams, and leaf 2 went from say 4.8 grams to 1.8 grams. So leaf 1 lost 2 grams, leaf 2 lost about 2 grams, more than one, nearly 3 grams. Which conclusions could be correct? So always do this, when you read a question, put some numbers on the x-axis and the y-axis if there are no numbers on it. Which conclusions could be correct? The loss of mass is mainly due to evaporation of water. Most matter close in both leaves after 1 hour. Yeah, well, let's look at the figure from 1 hour and then sort of levels out. So you have this point 1 hour. And leaf 1 has a larger surface area than leaf 2. Leaf 1 has a larger surface area than leaf 2. And leaf 1 has a thicker cuticle than leaf 2. Which conclusions could be correct? So the answer was A, 1, 2, 3, and 4. Then question number 12. Which statement about the transport of sucrose is correct? ATP synthesized by mitochondria of companion cells provide the energy to move sucrose from phloem sieve tube elements to leaves to sinks. The answer B was correct because it makes sense. Sucrose moves through plasmodesmata by passive diffusion from mesophyll cells through the companion cells to the phloem sieve tube elements. So B was correct, the rest are all wrong. The water potential in the phloem sieve tube element becomes more negative following the entry of sucrose. When water enters, then the water will equalize that water potential. Water enters companion cells and hydrostatic pressure builds up to push sucrose into the phloem sieve. No, it is actually the companion cells is by co-transport that sucrose enters, and then through the plasmodesmata it enters the sieve tube. What is the role, question 13, what is the role of the Casparian strip? Now, as you say, the role of the Casparian strip is what? It is to prevent water moving back into the cortex from the xylem. So the Casparian strip is that layer of suberin around the layer of endodermis cells. So basically, it actually acts as some sort of a prevention, because we want the root hair cells are too many, and as you see, a very, very old tree, the surface area of the roots increases a lot, and then of course too much water will be entering, and then a little amount of water is allowed to go ahead and into the xylem and up the trunk of the tree. Question 14. Sucrose is loaded into companion cells and phloem sieve tube elements by active transport and diffusion using transport proteins and plasmodesmata. So which row shows how sucrose is loaded? Active transport into companion cells, yes, that was correct. Diffusion into phloem sieve tube element, yes. Transport proteins would be in the companion cell, which is the hydrogen ions for co-transporting, and then the sucrose along with it coming back in. And plasmodesmata would be between the, say if this is the companion, this is the phloem sieve, then the companion cells are here and the plasmodesmata are here between the companion cell and this. So the plasmodesmata are somewhere here between the companion cell and this. I'll just show you a diagram of this. Then question 15. Which changes to the water potential and the volume of liquid in the phloem occurs when sucrose is transferred from leaves into phloem sieve to be transported to the sick? So naturally, if we've done this in a previous question, it was amino acids. So sucrose, when it's loaded, of course, will become more negative, and this will result in more water entering, and of course, the volume will increase. Same questions come again. The diagram shows the results of an experiment using leaves with the same surface area from two different species. So same surface area. Each leaf was left on a balance in daylight in a closed room and their mass recorded. So leaf X, what is the answer? Leaf X lost less water, and so less mass loss decreased, and leaf Y had more loss. So each leaf was on a balance in the daylights, and they result same surface area from two different species. Surface area means just the surface area. So they were both the same surface area. Maybe the one was longer or one was broader or something. Surface area was the same, but they were different species. So which feature of leaf X could explain that it lost less water? The mass was less decrease in mass. So the answer was C. It had sunk in stomata and an ability to roll the leaf, so there was less water loss. So that is how we know the answer was 2 and 3, because leaf X, say the mass was 5, it came down to 3, but in this, say the mass was something nearly 5 and it came down to 2 or maybe even 1. So lost 4 grams, and that lost only 2 grams. So why would you lose more water? Why would you lose less water? Less water would be because of the sunken stomata and the ability to roll the leaf. Now coming to question number 17, which features of these cells make them suitable for their function? So companion cells basically have to co-transport. So hydrogen ions have to be pumped out by active transport. So if you need active transport, you need numerous mitochondria. And then xylem vessel, how is it well suited for its function is the absence of cytoplasm, which allows mass flow. So that's why the answer was D. What is the sequence of events in the translocation of sucrose? Active loading of sucrose into sieve elements at the source, increased hydrostatic pressure, mass flow, and unloading at the sink. Then question number 19, where does water evaporate from during transpiration? Naturally, the surface of the spongy mesophyll cells. Evaporation takes place when the water vapors diffuse out of the stoma, which is through, not through the guard cells, but in the space between the guard cells. So this is how evaporation would occur in the surfaces of the spongy mesophyll cells from a liquid state to a gaseous state. So liquid to gas is evaporation, and then movement of water vapors, that means diffusion of water vapors out through this stomatal opening, is called transpiration. Now, coming to question number 20, which changes to the water potential and the volume of liquid in the phloem occurs when amino acids are taken out of a sink in a phloem sieve tube? Now, when they're taken out of a sink, the amino acids, when they are taken out, would of course take out all the solute. So this would lower the water potential. So solute, but it's saying changes the water potential and volume of the liquid in the phloem when amino acids are taken out of a sink in a phloem sieve tube. So the water potential will be lowered and the volume will increase. Then the diagram shows the transverse section of a stem, which area is the phloem. Now you know these sort of the very large ones, these are always the phloem. So this area would always be the phloem. And then of course there's the area, middle area, which is the cambium, and then of course the sorry, this would be the xylem, because the xylem vessels are huge and they have these hexagonal sort of an appearance. So this would be the xylem. So if this is the xylem, then D is wrong, and C would be the cambium, and then B would be the phloem. So you've got to be really able to understand photomicrographs. A leafy shoot was attached to a potometer and exposed to a variety of conditions. The time taken for the meniscus to move 50 millimeter along the capillary tubing was recorded for each set of conditions. So we've got all these different temperatures, humidity, air movement, and time taken to move 50 millimeter. Now this was the point that you have to look at very carefully. Time taken to move 50 millimeter. It moved 50 millimeter in how many seconds? 125 seconds, 71, 40, 166, 83, and 55 seconds, which shows the slowest and the fastest rate. Now, the slowest would be which was taking the longest time to move 50 millimeters. So that was 166. So 50 divided by 166.3. And then the shortest was 40. So it took only 40 seconds to move 50 millimeter. So that was the shortest time. So 50 over 40 will be 1.3, and that's why the answer was A. Please understand, I've done the working here for you all to understand this. Now looking at question number 23, the diagram shows 4 plant cells. Now you can see the plant cells which have a smaller vacuole, or the ones which are not plasmolized. You see, this was plasmolized, this was plasmolized. This one has a larger vacuole. 3 has a larger vacuole. This has a slightly smaller vacuole. Now the movement can only be from one which is fully turgid to the ones which are plasmolized, because it says in which direction could there be a net movement of water by osmosis. So 1 to 2 and 1 to 4 was the only possible. If you look at all the other threes, 1 to 3, that's not possible. This is a larger vacuole. Then 2 to 1 was also not possible. 2 is already plasmolized. And then 4 to 1 was also wrong. So you had to see the ones which were totally wrong, and that was the one that was all wrong, and you had to go on to the answer. 24. How will the root pressure in a plant be affected by waterlogged soils? Waterlogged soil means there's a lot of water, standing water, and there's no air spaces in the soil. Now, the answer was that why? If you look at the exam report also says in order for root pressure to form, cells surrounding the xylem actively secrete mineral ions into the xylem. Active secretion requires ATP from mitochondria, which in turn require oxygen to function efficiently. So root pressure would be affected by it because there will be no oxygen. No oxygen, so oxygen diffuses in through the air spaces in the soil. And if there's water standing in the air spaces, there's no air space at all, and there's just water because the soil is waterlogged. Then coming on to question number 25. Coming on to question number 25, the diagram shows the relationships. Now, in this, you have got to understand, you have to know the structure of the companion cells, the phloem sieve tube elements, and the xylem vessel elements. Now, you know that nucleus is absent in both. So nucleus is absent in phloem as well as xylem, while the endoplasmic reticulum is still present in the phloem and the companion cells. So that was the key point in which you had to understand what was missing in both and what was present in the companion cells. And so comparison was between companion cell, phloem sieve, and xylem. Another video will follow on the rest of the questions, and you need to revise this chapter before you do the MCQs. And another video will follow on some more questions on this chapter. And thank you for watching, and thank you very much.